Welcome to Botany class. Children, you have completed first 5 chapters. Let's begin the 6th chapter. The 6th chapter is the unit of life. Before entering into the chapter, you are going to study a topic, microscopy. You all familiar with this word microscopy, and you all know what is microscope, what's the use of it. You supposed to be seeing the microscope in your lab while you are doing your 10th standard practicals. From the lower class itself, you know what's the uses of this microscope. Let's see the functions of microscope in brief today. There are different types of microscopes. What we are using in the common microscope in our lab is the compound microscope or light microscope. Light microscope or compound microscope. Which we can say as bright field microscope. The first type of microscope is bright field microscope, dark field microscope, dark field microscope, phase contrast microscope, and electron microscope. Electron microscope. There are 4 different types of microscope given in your book: bright field microscope, dark field microscope, phase contrast microscope, and electron microscope. Today you are going to learn the first 2 microscope, that's bright field microscope and dark field microscope. Microscope, it's an inevitable instrument used for the studying of the cells and its subcellular structures. You know that the cells we cannot see through our naked eye because our naked eye has limitation in power. There is a limitation of human eye. We cannot see certain small objects beyond the limit of this size. We cannot see it, observe it. So we are using the microscope which magnifies the image, because the microscope which works on lens system. The lens system works there, which magnifies, number of lenses are used in the microscope which magnifies the image, and that's why we are looking it in an easy manner. It is visible because it magnifies the image. The compound microscope or this light microscope was invented by Isaac Janssen. Janssen was the person who invented the compound microscope. The microscope which works in number of lens system. So microscope is working with lens system. The properties of lens system and the light. Light is very important because at first we focus the object with the source of light. The image is magnified hundreds of times and it is visible to see our naked eye. So this image, the object, you may see there how we can mount the slide and how it is focused on the microscope. You may have the experiences to focusing the slides, objects on the slide and observing this object. Let's see this bright field microscope or this light microscope, which is the common microscope. Almost all the cell structures, all the objects, structure of ovule, or all these structures we commonly use to identify under this type of microscope. So this microscope functions as the image is directly focused by light. The source of light we use to focus towards the light. So this microscope here, this is the diagram of the ray diagram, how this light path. Before that, this microscope has the properties of light and this lenses has three properties, that is reflection, magnification and numerical aperture, NA, numerical aperture. So magnification, resolution as well as the numerical aperture. So what is meant by resolution? Resolution. The resolution, the term resolving power. Resolution is otherwise called as resolving power. The resolving power of an eye, human eye, is limited. We cannot see all the small objects. There is a limit to our resolution power of our eye. Beyond this limit, we cannot see. If the object is too small, some microns, millimicrons, we cannot see it through our naked eye. So that is resolution or resolving power. So this refers to the ability of the lenses to show the detail of the object present between two different points, between 2 points. If I am putting a dot here, you may see if you are very close to this, you can identify this. But if I am drawing two dots together, you cannot distinguish for distance. If you come closer, how much resolution power your eye has, you can identify. Here there is 2, or else you may think this one dot, that is resolution power, resolving power. So beyond this limit, we cannot see the object. So resolution is calculated by 0.61 lambda divided by NA, numerical aperture. So what is meant by numerical aperture? The numerical aperture is an important optical constant which is associated with the optical lenses. It's the important optical constant. It associated with the optical lenses which denoting the resolve, that is numerical aperture. Then magnification. Magnification which magnifies the image. So magnification is calculated by size of image seen with the microscope divided by the size of image seen with normal eye. Magnification is equal to size of image seen with the microscope divided by the size of image seen with normal eye. Now let us see this bright field microscope. This is dark field microscope. This is bright field microscope. The bright field microscope, you think about the normal microscope what you used in the 10th standard. There some slides, you know that the slides are microscopic slides which contains a specimen which is mounted by a cover slip. So this is the slide, microscopic slide. This is the object, a very small object. You may see there is a blue color or violet color or pink color dot. Only the dot you may see, but what it is you cannot see through our naked eye. When you are looking the slide, you may not see. But if you keep it under this microscope, you adjust the lenses and it is visible. How? At first, the microscope which contains number of lenses. Here is the object. We are placing the object in a stage. There is a place called stage. There is a adjustable platform present on the microscope. We can adjust it and we are fix the microscopic slide on the stage. So that is the adjustable platform. There at first we focus the light. So in this microscope, focusing the light directly to the object, and the object, from the object it shows the image through the different lenses. At first we are adjusting the condenser lens. There are number of lenses are there. One is objective lens. This lens is objective lens. Below this object there is a lens called condenser lens, and here there is a, near the eye there is a lens, that is eye lens. The different types of lenses we are using, these all lenses magnifies the images. So in the objective lenses, objective lens which magnifies, there are 4 different types of this lenses: 5x, 10x, 45x and 100x. Different lenses are there. So at first the condenser lens which focus the source of light, direct light can be focused directly to the object. That's why first we are adjusting the lenses which focus the object. So this lens, it makes the image and it is fall to the eye lens. Near the eye there is a lens. So this lens which magnifies, these 2 lenses which magnifies the image and it shows as a visible object through our eye. So when you are looking this through our eye, now this image is visible because these lenses are magnifies the image 100 of times. So which magnifies and gives the image, that's why it's visible to our naked eye. Then if you see this dark field microscope, that is light microscope, bright field microscope, directly focus on the object. The light is focused directly to the object, and at first there are 2 different images it forms, the primary magnification and the secondary magnification. Primary magnification from the objective lens. First the objective lens magnifies the image, that image is called as primary magnification. Primary magnification. And this eye lens, that is eyepiece, we used to say that eyepiece which magnifies the image, secondary image, that is secondary magnification. So 2 times it magnifies the image, and when you are looking here through the eye, naked eye, the image is visible. Final image, it becomes an inverted image. So it inverts the object and it shows the image clearly to see through, it's visible through our naked eye. Let's see this dark field microscope. So first type of microscope is bright field microscope. Draw this diagram and explain how it works. There are 2 different lenses which magnifies primary image as well as secondary image. Now dark field microscope. In dark field microscope, the normal magnification, the normal lenses which contains a special type of device, that is called patch stop carrier. Here the important point is patch stop carrier. This is called as patch stop carrier, which is nothing but a glass device. It's a glass device that is called patch stop carrier. So here the source of light, the source of light may be the compact fluorescent light or light emitting diode. So compact fluorescent light or light emitting diode is the source of image. So in this microscope, the field is dark, but the object is glistening. Field is dark. The name of the microscope is dark field microscope. Here the field is dark, In the dark, the object is glistening. In the dark field, the object is glistening. So, here there is a patch stop carrier, which is the glass device. In this glass device, it contains a central patch. In the center region, there is a patch, and the margin and the center, it leaves a small opening. So, this source of light enters through this patch stop carrier. That's a device. So, through this device, when the light passes, it forms as a, here there is a condenser lens. This lens is called as condenser lens. Now, this condenser lens, when this light rays are passing here through this opening or pore, small pore throughout the margin in the glass device is there. Through this, the light passes and it focuses on the condenser lens, and the light goes as a hollow cone. See, this is a hollow cone. It goes and the object is present here. This is the stage, adjustable stage, adjustable the platform which is called the stage. Here the object is present. So, in this object, when the light reflects here, this again it focuses to the objective lens. At first there is a condenser lens and here there is a objective lens. So, it focuses the images in the objective lenses. So, through this dark field, the object is glistening now. So, simple method of this dark field microscope, which is mentioned by Z. Zsigmondy. Zsigmondy, who explained this dark field microscope. So, two microscope today you learned. Number 1, bright field microscope. Number 2, dark field microscope. So, what are the questions you are going today? First, what is meant by resolution? What is magnification? What is resolution formula? Magnification formula. Then 5-mark question. Then numerical aperture. Then there is 2 questions, bright field microscope and dark field microscope. Draw these 2 diagrams in the class note. Then 2 more types of microscope is there, phase contrast microscope and electron microscope. Let us see in the next class. Thank you.